Hello. We know that most molecules absorb light at different wavelengths though. And we know some molecules like chlorophyll and beta carotenes which absorb light and allows the sustenance of life in diverse forms like animals on earth. Similarly, DNA and RNA also, or nucleic acids, proteins, they also absorb light. And that can be used to estimate many things or the forms of the DNA as well. I am Dr. BCMR, Assistant Professor at Shastra. In this discussion, we will talk about the absorbance of light and how we can use it as a principle to estimate the structure of DNA. We know that DNA absorbs mostly or maximum at 260 nanometers. In this case, we will compare with that of DNA and say proteins. DNA absorb at maximum at 260 nanometers and proteins absorb at 280 nanometers. The absorbance is mostly because of the planar heterocyclic ring structures of the bases and their resonance between the different forms they may have. In proteins too, it is still the ring structures like tyrosine, tryptophan, and phenylalanine. So the absorbance, in case of DNA, the deoxyribose and phosphate, the backbone is not playing as much role. It's majority of it, at least in absorbance at 260 nanometers, is because of these planar structures of the nitrogenous bases. To have it as a summary, it is a nice post I picked from the Bubbling Biochemist, and here is the absorption spectra and of different compounds. At 260 nanometers, it is nucleic acids, that is these bases absorb at 260 nanometers. At 280 nanometers, we have the tryptophan, phenylalanine, and tyrosine that absorb at 280 nanometers. So we can use, say, if we are trying to purify DNA and we want to estimate how pure it is, we can always have the comparison between 260 by 280 nanometers ratio. Usually that ratio is going to play a big role, and if it is above something like 1.8, it is around 1.8, then it is considered pure enough for laboratory experiments. And if it is, and there are also other components that we do use, such as the salts and phenol, they may have absorbance as 230 nanometers as well, and because of which can be used to estimate the purity of DNA that we purified in the lab. And today, what we will discuss is about trying to understand the structure of DNA, or the being from single-stranded to double-stranded, and how it can be done. There is a phenomenon which we will call which is called as the hyperchromicity. Hyperchromicity, as it say, hyper is more, and chromicity, in this case, it's about absorbance and increase in absorbance of at 260 nanometers. So for DNA and RNA, we use 260 nanometers. Say in the case of double-stranded DNA, the bases are stacked something like this, and they are in pretty stable form, and they are not absorbing the 260 nanometer. Their absorbance at 260 nanometers is lesser compared to a single-stranded DNA, for example, where the bases are much more available in a way. So if we take a double-stranded DNA, and its absorbance here is likely to be somewhere around there, it is little lesser. And if we are increasing the temperature, that means we are breaking more number of hydrogen bonds, the DNA will slowly start denaturing. And then when it comes to single-stranded DNA, when it becomes single-stranded forms, then the absorbance is more. So if we talk in terms of the compounds or the chemical composition in this state and the chemical composition at this state is same. What is different though is that in one case here it is double-stranded, whereas in the other case it is single-stranded. And what we see because of that is this huge increase in the absorbance. This is the amount of change in the absorbance at 260 nanometers. That increased absorbance is referred to as hyperchromicity or hyperchromic effect. Meaning in our situation here, we are using it to study the transition between double-stranded to single-stranded DNA. So when we are estimating different DNA based on their base composition or the solutions in which they are present, will have difference in the hyperchromicity. To have an accurate value or to designate a value to the nucleic acid or the DNA or genome, for example, we use a point called as Tm, which means it is the melting temperature. It is actually the midpoint. It's actually the midpoint in which we we take this as the melting temperature at which half of the DNA that we have taken is in single-stranded DNA and the other one is in double-stranded. So we could have taken this, but there is no exact point where we can determine because of the limitations of the equipment that we use. We can also not be very accurate in picking this one. Therefore, it is a good thing or nice way of, which is a much more accurate reflection of a within the considerations of the equipment we use, the spectrophotometer. So the midpoint is called as the Tm, and that is the point that it is the temperature at which the DNA, half of the DNA in the sample is in single-stranded and the other is in double-stranded. Tm is usually characteristic of the base composition. It could also vary based on the conditions we use. Like in the previous discussion, we spoke about 2 or 3 important factors. One of it is GC contents. The other one is about salt concentration, how they can affect the structure, affect the stability of the double-stranded DNA. If we can recollect, higher the GC content, the longer they are likely to be, they are in different conditions, they are likely to be together. In this particular representation, we see 3 different samples. We'll call them as A, B, C. 3 different samples. Assume that the number of bases is equal, number of base pairs is equal, and what is different in them among these A, B, C DNA samples is that the GC content. If GC content is higher, then we can expect more hydrogen bonds. That means they require more, they will be together, the 2 strands will be together for longer, or you need more energy to break them up. Energy in the form of heat here. So if we see the DNA which has higher GC content is slowest to denature. So the midpoint we will consider just like this approximately. This is the midpoint, and so is here and here. If you see that A, which has the less amount of GC content, is likely to denature faster than that of C sample, which has about 60% GC content. We also can have discussed about salt concentration, and we discussed about what is the role of sodium ions that they play, They are likely to reduce the negative charges by binding to DNA on the phosphate backbone, because of which they reduce the negativity or reduce the repulsive forces between the two strands. So they are likely to be more. So in this experimental result, we have 0.02 molar NaCl, and we can compare it with 0.6 molar NaCl solutions. The one with lowest amount of salt concentration is likely to denature faster compared to that of one in higher concentration. So these are experimentally proven, and I hope we can remember the logic of it. So we have discussed about hyperchromicity, which is the increase in absorbance upon transition from double-stranded to single-stranded DNA. We'll discuss about melting temperature, and that is the midpoint at which the nucleic acids or DNA, half of the DNA in the sample is in single-stranded and the other one is in double-stranded. We also discussed about absorbance. Nucleic acids absorb at 260 nanometers because of the heterocyclic structures of the bases, and proteins absorb maximum at 280 nanometers because of tryptophan, tyrosine, and phenylalanine. Say, for example, if there is a protein that is low on these three aromatic amino acids, that is likely to have very reduced absorption. Thank you very much.